So again, since this material is essentially up there, both from me talking about it as well as other professors at different institutions giving the same sort of overview, I'm going to go through this fairly quickly. What did it do? There it is. Okay. So there's kind of two halves to chapter 2. There's a part that talks about Mendelian inheritance, the transmission, some of the experiments that he went through, and how that deduces down to the 2 laws or principles that we attribute to Mendel. And then there's another part that links into the statistical analysis that we use in genetics to make predictions of outcomes for crosses. And so we kind of address those separately. So the first problem set deals essentially just with Mendelian inheritance, and the next problem set will come in, we'll talk about statistical analyses. And we'll also hit that in our first lab coming up next week. So there are multiple ways of kind of stating the genetic principles that Mendel came up with. This is one particular way. As we progress through this slide, I'm going to have you write down a precise definition I want you to memorize for the law of segregation and the law of independent assortment. Okay, but essentially, Mendel figured out that each trait was controlled by a pair of alleles. He called them factors because he didn't have the word allele yet, but 2 alleles, and those alleles must be separated somehow during reproduction. Okay, we know how. They're separated through the process of meiosis, and then each allele is going to end up in a gamete. That gamete is going to be the starting point of the next generation, so the offspring. Okay, and he saw that had to be happening. What he also saw was that for one particular trait, one of the versions was passed on, but for another trait, another version for that trait would be passed on, and they could recombine in any possible way. So if you had 2 traits for height and 2 traits for color, you could get 4 combinations in the offspring. And he deduced that this was happening because the inheritance or the transmission of one trait is independent of the transmission of the second trait's allele. Okay, and there's a better working definition of this on the slides ahead. So the way Mendel did this was by mating organisms, right? Performing what we would call a genetic cross. Okay. When he did this, he was creating organisms that had a recombination or a rejoining of 2 different sets of original traits. And this is called a hybrid. Okay. The offspring that result from a blending or a mixing of traits from 2 different parents. So the cross itself can be referred to as a cross, which it usually is. It can be referred to as a mating. It can be referred to as a hybridization. Hybridization kind of caught on in the plant world. With animals, we call it breeding or mating, okay? But in both cases, you're taking individuals with different characteristics for a trait, mating them, producing offspring, and examining what the characteristics of the phenotype are in the offspring, okay? Now, what many people had observed around in Mendel's contemporaries was that when you mated a couple of different critters or plants, you got essentially a blending of many of their traits. And people were looking pretty much at the whole organism in total. Well, what Mendel did was he focused in on just one particular trait at a time. And by doing that, he realized that for each individual little component of the phenotype, they either look like one parent or the other parent. They were not blended, okay? So if we take a trait like yellow and green, which were 2 of the colors of the seeds were just yellow or they were just green. They weren't an in-between yellow-green color. They were identical, or they were tall or short. They weren't medium, okay, than the plant height. So it wasn't blending. So that was a key component of his particular analysis that many other geneticists, or not really geneticists, but people doing these kinds of experiments had kind of missed. And because of this, this is how Mendel was able to come up with the fact that each of these particular traits must be being controlled by two factors, okay, two alleles, and those alleles were being segregated during the reproductive process, we know, meiosis, segregated during meiosis, and then recombined during fertilization, and you could pass on the traits in pretty much any combination. So the first thing that is usually necessary to do sort of a breeding experiment is to start with organisms that you know are always going to be the same. This is known as a purebred or a true breeding organism. What it means is, if you had a Chihuahua that was a purebred Chihuahua, you had a boy and a girl Chihuahua, and you mated them, what are you going to get? Chihuahuas. And if you take those puppies and you take the boy and girl puppies and you mate them, what are you going to get? Chihuahuas every single time, okay? So they are pure breeding. And we use terms like a strain, if we're talking about maybe microorganisms. We use breed, if you're talking about, say, a breed of dog. Agrivar, if you're talking about a particular kind of crop. And maybe variety, if you're talking about a particular kind of flower. But what it means for all of these is that the only version of the gene they have is the two alleles of every trait they have are identical. That means they're completely homozygous, or nearly so, okay? Now, when you have an organism like that, there's going to be no variability in their offspring. So if you take any member of that strain and mate it with any member of a different strain that's also homozygous, you're now doing a cross, okay? So what Mendel did first, of course, he worked with pea plants, but what he did first was he picked out one trait that he would examine, and he would cross two different pure breeding strains that were different for that trait. And he had 7 different things in particular that he discovered followed the pattern. Okay, and so these would be called a monohybrid, because you're looking at one trait, you're looking at 2 versions of that trait, and you're examining what happens in the phenotypes of the offspring. And in this cross, you will follow it for 2 generations. You will mate a pair of parents. They will produce children. You will then mate the children to produce what would be grandchildren if we were talking human. Now, a big difference between doing this with animals and doing it with plants. With animals, you have to have 2 different individuals, okay, for anything you would want to ever really do. So you have a parent, and it's going to mate with another parent, okay? So that's called the parental generation. This is going to produce the first generation of offspring. They're going to be called the F1 generation, okay? You're not going to have just one, you're going to have lots, okay? You're going to have males and females if they're animals. You're going to have plants, which generally could fertilize themselves, okay, because they contain both male and female parts. And if you let the F1 offspring mate with another F1 offspring, okay? So if this is plants, we would call it a self-cross. because it's just pollinating its own flowers. If this were animals, we might call it a sib cross, because they are going to be siblings that cross with each other, okay? And from this, you're going to produce a second generation of offspring. We would call it the F2 generation. And it's the proportions of these, okay? These typically will all look the same. Okay? All the members of the F1 generation will look the same phenotype. But here you will have multiple phenotypes. And there will be some proportion of different ones, right? So there will be distinct ratios of the different phenotypes. So this is what Mendel's experiments did, which other people had never done, and that's why he figured it out, okay? So what I drew on the board here, it is with prettier pictures. If you had two different phenotypes of parents differing by their height, and you make them cross-fertilize them, okay? You produce the seeds, the F1 generation grows up, and they are all the same height. They're all tall, actually. You let them fertilize their own flowers, they produce seeds, you plant those seeds, and now there are mostly tall plants, but there are also short plants again. There are no plants that are medium, okay? A lot of people are confused. They're like, so how tall is a tall plant and how short is a short plant, okay? The tall plants actually form vines, 12 feet tall vines that would vine up a pole. The short plants do not do that. They only grow to be about 1.5 feet or 2 foot tall. So very different, very obviously different. They don't make a vine. Well, here you find most of them make a vine, but some of them don't. And Mendel would do that for multiple different traits, and we'll look at them in a minute. And each time he saw the same trend. He saw, you take two traits, I don't care what they are, the first generation looks like one of the two parents, only one of the two parents. It's either tall or short, or it's green or it's yellow or whatever, but it only looks like one. The second generation, both phenotypes show up again, okay? Now, again, with animals, you can't do the self-fertilization thing. So you have a couple of parent generations. They're going to have some offspring, right? Then you're going to have to just mate with another offspring of a different sex, but of the same generation, okay? So, and then you're going to have a second generation after that, right? And you're going to have these phenotypes being passed down. That's just for fun, okay? So what did Mendel do? He saw 7 different things he looked at, and he noticed that every time in that first generation, the F1, all organisms were identical, and they had only one or the other of the two parent traits. So if you had yellow and green seeds, here, only yellow, okay? But in the second generation, you saw yellow and green again, not equal numbers, but in a proportion. And if you look at that here for the flower, where the flowers are, or the color of the pods, or the shape of the seeds, or the height of the plant, he saw that happen again and again. And he just said, well, what's the ratio here? And the ratio turned out to be almost the same every time, nearly 3 to 1. He always saw 3 of one of the traits, the trait that showed up first, and then he would see 1 to every 1 of the second trait. So this is how he came up with the idea of dominant and recessive. So here's a definition. The dominant trait is that trait which appears in the F1 generation. of a monohybrid cross. It's the trait which appears as the F1 phenotype in a monohybrid cross. Okay. Now there's a difference between the dominant trait and the dominant allele. Okay. So the dominant allele is the version of the gene, which dictates the phenotype of a heterozygote. The version of a gene which dictates the phenotype of a heterozygote. The heterozygote has to have two different alleles, and one of them is the dominant allele, and therefore that one allele says that the organism has the dominant trait. Okay, so there's a connection between dominant alleles and dominant traits. To have the dominant trait, you should have the dominant allele. All right, so in the F2 generation, he saw this ratio of 3:1. Okay, so he proposed that for every trait, there has to be two versions, alleles, right? And during reproduction, during meiosis, one of those alleles is passed on from one parent, and the other allele, and it might be the same or it might be different, is passed on from the other parent. Okay, and so the parents might be homozygous or they might be heterozygous. And if they're homozygous, then they're only going to pass on one version of the allele, and every offspring would look the same. That's pure breeding. But if they're heterozygous, then they will pass on a dominant or a recessive. And the likelihood of passing that on is 50-50. They have one of each, right? You have two coins in your pocket, right? They're both quarters. One's dated 1965. The other is dated 1972. What's the probability when you reach in your pocket of picking up either of those coins? You're only going to pull out one. 50-50, right? You see, if you have two choices, the chances of making one of those two choices is 50-50. So there's a 50% probability that the dominant one is passed on. There's a 50% probability that the recessive one would be passed on if you're heterozygous. If you pass on the dominant one, its effect will be seen in the phenotype. If you pass on the recessive one, its effect is not seen. We say it's masked. Okay. So the recessive trait is the trait which does not appear in the F1 generation. So in genetics, we don't draw the pictures of the plants or the flies or the people. We simply write down a symbolism to represent the alleles, to represent the genotypes of the alleles and the genotypes of the F1 and the F2 individuals. And then based on our understanding of the genes, we can deduce their phenotypes. So in the initial parents, you would have had two traits for tall, for being tall. And you would have two alleles, sorry, I said traits, two alleles, I meant, for being tall, and two alleles in the other parent for being short, okay? So most of the time we pick a symbol that designates something about the trait. So T for tall. And if you have two versions, the dominant version of that trait is generally given an uppercase letter, while the recessive version is often given a lowercase letter. This is not a universal rule, but it's it's close to universal, okay? Most of the time, they're not given just a single letter. Many symbols consist of multiple letters and numbers, okay? But this plant is going to pass on one allele. So what are the choices? There is no choice. It only has the dominant allele because it's homozygous. And this plant has no choice. It may only pass on a recessive allele because it is homozygous. And those two gametes would fuse together, the fertilization process, to make all of the progeny. Every time that happens, you get a new progeny, and all of the progeny then are heterozygous. Okay? So the progeny of an monohybrid cross are heterozygotes. They're all going to be heterozygotes if the two parents start out with different traits. Now, if these are heterozygotes, do they show both traits? Are they tall and short at the same time? Now, see, that's the kind of thing about a Mendelian trait. You only have a choice of being one way or the other, right? They're mutually exclusive. You can't be tall and short at the same time, right? Like your thumb. You can't be on the left on top and the right on top at the same time. You could only have one choice or the other, right? So which of the two traits is it? It's tall. Tall's dominant, okay? So at first you might not know whether or not you should write a T, capital T or a lowercase t, but once you do the experiment and get the data, you know. Tall is dominant. So that plant is heterozygous. What happens when those plants produce gametes? Well, if we follow here and we take this genotype, we can see that you could pass on a big T. Have it bigger on the next slide. You could pass on a big T. Each parent could 50-50 chance pass on a big T. So some of the progeny are going to get both big T's. You also have a 50% chance of passing on a little t, while the other parent passes on a big T. And you've got two ways that could happen. It could be the little from parent one and the big from parent two, or it could be the big from parent one and the little from parent two. So that could happen. There's two possible ways that could happen. So it's going to happen twice as often. And then there's one last way where each parent could pass on only their recessive T allele. So there are 4 different combinations that could exist, right? And if we look at those combinations, there's really 3 different things there. There's being homozygous tall, there's being heterozygous, or there's being homozygous recessive. Now, did Mendel observe 3 different types of offspring in his experiment? No. What did he have? That's right. How many did he observe? 2. So what that means is some of these genotypes have to be giving the same exact phenotype, right? Well, we know it. That's going to be tall. We know that's going to be the short ones. So the question is, well, what were these? Well, did you see more tall ones or more short ones? More tall ones, 3-to-1, right? So that means that just like up there, those were tall. These are also tall. So if we take it, there's 3 ways to be tall and only 1 genotypic way to be short. So when we look at the genotype, we can see, yep, there are 3 different genotypes. And that would give us a ratio of 1 to 2 to 1. That's called a monohybrid genotype ratio. Any monohybrid cross will produce a 1 to 2 to 1 F2 genotype ratio. Okay. You don't have to know what the trait does. You don't need to know how it's inherited or anything. You just need to know that when you look at the genotypes, there are going to be 3 different versions: a homozygous dominant, a heterozygous, and a homozygous recessive. Okay. Now, when you look at the phenotype, this is where it comes and becomes more complicated. What Mendel observed were traits where the heterozygote produced identical phenotype to the homozygous dominant. We call this mode of inheritance complete dominance. We call it several things. Complete dominance is one. That's the one I usually use. Complete dominance. That means that that dominant version of the gene has absolute control. One copy has absolute control on the phenotype. Okay? It's also known as simple. It's also known as Mendelian. All inheritance is Mendelian, so that one's not really right, but it gets called those things. Okay, so don't worry if you hear it, they'll probably mean the same thing. Now, in that situation, in the case of simple dominance, the phenotype ratio will be 3:1. These three genotypes will produce identical phenotypes. And this one genotype will produce a separate recessive phenotype. So the ratio will be 3 dominant to 1 recessive. Okay? That's called a monohybrid simple dominant phenotype ratio, or complete dominant phenotype ratio. Are there other phenotype ratios that could happen? Can you guess? For this, are there other possible ratios that could happen? In other words, does this genotype have to give exactly the same phenotype as this, or vice versa? Does this genotype have to give a tall? Does a heterozygote always have to look like the dominant homozygote?
Not always.
No, not always. And in that case, there's another mode of inheritance. We'll jump into a little bit more detail later, but we call that mode intermediate. Okay, intermediate. It also goes by incomplete. I don't like incomplete because it makes it sound like you didn't get everything, but you did. It's just not working. Okay, so it's intermediate. It's in between. In peas, height is not intermediate. Okay, but in some organisms it is. And so what would happen is you take a tall thing and a short thing and you hybridize them, and your F1s would all be medium-sized things. See how that's different? What did Mendel see? He took a tall and a short, and what did he get? All tall, right? But what if you took a tall and a short and you got all medium? Well, that's blending, and that's intermediate. Let's say you took those two medium ones and you let them mate. What would you get? And what variation would you get?
Tall, short, and medium.
You'd get all three. You'd get tall ones and short ones and mediums. And how many of each kind do you think you'd get? Well, you'd get one tall one and two medium ones and one short one. With this, with complete dominance, the phenotype ratio is different from the genotype ratio. Okay? With this type, phenotype ratio is identical to the genotype ratio, and that's true with every other mode of inheritance. So the only time the phenotype and the genotype ratio are different is when there is simple dominance or complete dominance. Okay, well, This observation led Mendel to the formulation of the law of segregation. And that in blue down there is what you want to write down and memorize, okay? Every word is important. No words can be left out, okay? They mean something very specific. You can't substitute any words either, okay? Each. That means you have two, and you have to look at both of them. Each allele of a locus, that's a place on a chromosome. Every place on a chromosome is an allele. Each allele of a locus is going to be placed, because of meiosis, it's going to be placed into a separate gamete, individually from its other counterpart, okay? So each allele of a locus is placed individually into separate gametes, okay? For a given gene. For a second gene, the same thing happens. For a third gene, the same thing happens. For a fourth gene. So every gene, every locus on all the pairs of chromosomes, I mean, it's one chromosome is going to end up in one cell and the other chromosome is going to end up in the other gamete. So they're always going to be placed like that. And he deduced this because he could tell, hey, there has to be two of everything, because that's the only way to come up with a 1 to 2 to 1 genotype ratio. And from that, that's the only way to come up with a 3 to 1 phenotype ratio. So he worked this backwards from looking at that ratio. Okay. Now, what he didn't know about was meiosis. That's fine. But what he understood was something was happening. And it wasn't too many years after this that people figured out what was going on in meiosis. So that was okay. But what he could do, since he couldn't just see this happening, was he could make a prediction. And he could test that prediction. And that test is called a test cross. Okay? And since this is a monohybrid, therefore monohybrid test cross. But what he said was, look, I got 3 tall plants for every 1 short plant. And so I know that I must have 3 genotypes. I know 2 of those genotypes are heterozygotes, and one of them is a homozygote, right? He's like, so I need to test. If the ratio is really 2 heterozygotes for every 1 homozygote, right? That's what he figures. And all of these are tall. He says, well, what I know is the short guys, the short plants, have to make only short gametes. They can only pass on the short ones. Now, the tall ones, though, can do 2 different things, right? You have 2 different choices. So he said, what I'll do is I'll take a tall plant and I'll breed it with a short plant. I'll do it for all the tall plants. And I'll breed every tall plant, no matter what I think its genotype is. I'll breed them all to short plants. And I got 2 choices. If they're homozygous, then all the offspring are going to be tall. But if they're heterozygous, I have the possibility that sometimes I'm going to get heterozygotes. Those would be tall. And sometimes I'm going to get homozygous shorts, which will be short. So when you do this cross, you have what proportion of plants are going to be heterozygous. How many total kinds of plants are there? Add that up. 3. And how many out of 3 can be heterozygous? 2 out of 3. So 2 out of 3 times you should get half tall and 1/2 short. Right? And how many could be homozygous? 1 out of 3. So 1/3 of the time you should get all tall. Right? So you take all your tall plants, You breed them all to short plant and ask, how many times did I get only tall offspring, and how many times did I get tall and short offspring? And sure enough, 2/3 of his offspring, or 2/3 of those matings, look like this, and 1/3 of those matings gave him this. And so from that test, he could tell that in fact, the only real logical explanation was that being heterozygous makes you tall and being homozygous tall makes you tall. And that it only takes one allele to dictate the phenotype. And he ran with that, and because he did, he could figure out a lot of other things, like the law of independent assortment. Now, this day and age, we know that that's not always going to be true, because sometimes you're going to need 2 alleles to give a phenotype, and if you only have one functional allele, you get a different phenotype, okay? But when he noticed those kinds of things, which he probably did, he was like, so 7 out of 10 things follow this pattern. I'm going to kind of ignore those other 3 things. I'll figure that out later, okay? And because he was able to sort of focus in on the simple inheritance pattern, he figured out the rule. So he went on to the next stage of experiments, which was, okay, I know what's happening now for one trait at a time. What happens if I track 2 traits simultaneously in a mating? This would be a dihybrid cross. 2 traits are different. Let's say maybe the color of the seed and maybe the shape or the form, texture of the seed. And you could have 2 different possible inheritance patterns from this. He figured it like this. He said, maybe the dominant traits will always go together and the recessive traits will always stick together. So dominant will go with dominant, recessive will go with recessive. And he called that linked. That dominant would be linked to dominant, recessive would be linked to recessive. So that's a hypothesis we're going to call linked. The alternative would be that dominant color could go with recessive shape, and dominant shape could go with recessive color, and recessive could go with recessive, and dominant could go with dominant. So basically you can make any combination, right? So the other one would be any combination, and he called that independent assortment, okay? Somebody did. I don't know if Mendel did it or not, but some Yahoo did it. So here's what you're thinking. Here's what his mind was thinking was, all right, I got these yellow round peas. I know he knew yellow was dominant already, and he already knew round was dominant from the monohybrids. And he had some green wrinkly peas, right? Those are the peas you're probably familiar with because they're sweet, and that's what we eat. Those are the peas that most people eat. You don't usually eat the yellow round ones because the round ones aren't as sugary. They don't have as much sweetness to them, and they're also a little bit harder texture-wise when you soak them. So, but that's the dominant version, okay? So they make gametes. The only kind of gamete would be dominant trait, dominant allele. The only kind of gamete would be recessive alleles. So what's the F1 going to look like? So the F1 is going to carry a dominant recessive, a pair of dominants, and a pair of recessives. And we'll separate them like this so that the dominant ones are on one side and the recessive ones are on the other side. Okay. This might be a good time to bring up this little way we notate the genotypes. Okay. So you're going to have the 2 symbols. What do they represent? Well, they represent an allele, right? And so you've got 2 alleles together. When we write them like this, it represents a locus. So together, this represents a locus. And also, they each represent one of the homologs of a homologous pair of chromosomes. Okay? So we kind of really put a lot of representation into that one little symbol. So you have a pair of homologous chromosomes, right, with each homolog's allele represented on either side of a slash. Okay, so when we represent that with a slash, we mean everything on one side of that slash should be passed on in a gamete. And everything on the other side of that slash should be passed on in a separate gamete, right? So the things on each side of a slash are going to segregate. That's what the slash means. Here's the line. Everything over here is going to go in one gamete. Everything over here would go in a different gamete. Okay? That's the law of segregation. So what he's saying is, this thing is going to make gametes that are either YR or little yr. And that's it. That's its only options. And then when they fertilize, right, plants don't make sperm, but whatever. When they fertilize, pollen is what they should have wrote. When they fertilize, you get recombinations, right? There's 3 ways to get a dominant YR. One of those ways you get homozygous. 2 of those ways are heterozygous. Those should all be yellow and round. And then there's one way, and only one way, to be green wrinkled. You're never going to get yellow wrinkled. You're never going to get green round. There's just no way to get it. You can't put any of these together in any way to get anything other than those 2 phenotypes, yellow and green. And predictably, you should get 3 yellows for every 1 green. He's like, okay, that's kind of what I think is going to happen. Okay. What about the other possibility? The other possibility, you start off the same. Notice a huge difference in the way the genotypes have now been written. Okay. So what does this big difference mean? It's a huge difference. You're like, really? It's not that huge. It's a huge difference. Huge. Okay. It's going to be great. It's huge. All right. So don't call them the National Guard. Okay. So big Y's and big R's are not connected. They are on separate chromosomes from each other. And we can indicate that by throwing a little semicolon in between the loci. So the big Y / big Y is for one locus on a pair of homologous chromosomes, and the big R's are for the loci on a totally separate pair of homologous chromosomes, unlinked from each other, independent of one another. So now what can they make? As gametes, they still will make a gamete that carries one of the Y's and one of the R's. So all they got are capitals. They got dominance. All they got is recessive. So when those fuse, now we're going to be heterozygous for Y and heterozygous for R. Phenotypically, still yellow and round. But if we look now, since these are all independent, the gametes that can be made could get a big Y and a big R, or a big Y and a little r, or a little y and a big R, or a little y and a little r. There's really four possible arrangements that could occur, and they should theoretically be relatively the same. They should all happen with equal probability. And that would happen for both male and the female. So what happens if you have four possible eggs that could pair up and fuse with four possible sperm? That means you get a whole bunch of arrows. You get 16 possible outcomes. So you follow an arrow, go this one and then go there, this one and 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 there, and so on and so forth. Okay? 16 different things. And if you look at the different things, you get some that are round and yellow, we expected, some, but actually very few, that are wrinkled and green. But what do you get that we would never see here? You get wrinkled yellow and round green. Those two phenotypes should never appear unless everything could basically rearrange and mix with any other allele. So any allele from one gene could go together with either allele of a second gene. And that would be independent assortment. And in fact, that's what Mendel saw. Okay, here's his numbers. He saw 300, and he did this experiment. This is actual data, 315 dominant for both, 32 recessive for both, but he saw about equal numbers that were hybrid. They had one of the dominant traits along with the other recessive trait, and that would not predict it, okay? And when he put the ratio formula to it, it comes out 9 double dominant to 3 hybrid to 3 hybrid to 1 double recessive. And that ratio came up every time he did this. He did it about 3 times to satisfy that it was happening. It came up that way every time. So that means that this does not happen, okay? That independent assortment is happening, okay? And if we look at this, we get possible genotypes up here listed, okay? And then we can then put the genotypes down here by just calculating them all, writing them all down. And what you notice is there's only one that's like this, where everybody's dominant. There are 2 that are like this, where the R's are dominant, but the Y's are heterozygous. There are 4 where they're both heterozygous. There are 2 where the R's are heterozygous and the Y's are dominant, okay? But every one of those genotypes is going to give you the same outcome phenotypically, right? If you have one dominant allele, you get the dominant trait, right? That's what being simple dominant means. So if you have a big R and a big Y, guess what you get? Round and yellow, okay? There are 9 possible genotypes, 9 possible combinations that produce that. If we look at the recessive, there's only 1 possible combination of genotypes that allow you to be homozygous round, homozygous wrinkled, homozygous green, homozygous recessive for everything. There are 3 genotypes that would give you round green seeds and 3 genotypes that would give you wrinkled yellow seeds. Okay. So there are 16 possible genotype combinations, but if we look at those 16 genotypes, several of them are the same. So this gives us something, a genotype ratio right there. Okay. Now there's 2 ways to write it down. You can write it down the way it's written right there. That's fine. Or you can say it the way I say it to myself, because it just rolls better. For me, it may not for you, but I go 1, 2, 1, 2, 4, 2, 1, 2, 1. Right? You take the first one, 1, 2, 1, and double it. 2 times 1 is 2. 2 times 2 is 4. Right? 1, 2, 1, 2, 4, 2, 1, 2, 1. There are 9 different things. 1 thing, 2, 3, 4, 5, 6, 7, 8, 9. There are actually 9 distinct genotypes. But if you look at those, there's multiple ways to get each one. There's 4 ways to get that. 4 combinations can give this. So it should happen 4 times as often as this. Okay. Now, phenotypically, it's 1, 2, 4, 2. 1, 2, 1, 2, 1. So if you want to memorize it by phenotypic group, that's okay. Just throw a 2 and a 4 in there on the first group. 1, 2, 4, 2, 1, 2, 1, 2, 1. You just flip it over. All right. Is there some rule that could allow you to predict this for as many genes as you would like to predict it for? Yes, there is. Because perhaps you can see a pattern. 1, 2, 1 is the genotype ratio for what? Monohybrid. What is a dihybrid? A dihybrid is 2 simultaneous monohybrid crosses. Okay? So we can treat that mathematically. Genetics is just math, cleverly disguised as biology. So if you had a monohybrid ratio, you would get 1, right, homozygote, 2 heterozygotes, 1 homozygous recessive. How many total genotype types are there? Well, there's 1 like this, plus 2 heterozygotes, plus 1 homozygous recessive. So you just turn those to pluses. What if you had another one? So if this deals with yellow, this deals with round, okay? Now, what would you get if you happen to be yellow, homozygous, and round homozygous? What's the likelihood? Well, what's the likelihood of being homozygous? There's one way to be that way, right? How many total ways are there to be something? 4. So your ratio is now 1/4. There are 2 out of 4 ways to be a heterozygote. There is 1 out of 4 ways to be a recessive. So what's the probability of being a homozygote? 1/4. What's the probability of being a round homozygote? What's the probability of being round and yellow? 1/4 times 1/4, right? This is called the multiplication rule, or the product rule for genetics, or for statistics. 2 independent events could happen, right? What's the chances of being a boy or a girl? 50%. What's the chances of having your right thumb on top or your left thumb on top? 50%. So what's the chances of being a boy with his right thumb on top? Half times a half. Okay. So this is the product rule. If there's 2 independent events, if one thing doesn't depend on the other thing, then the probability of those 2 things happening simultaneously is the multiplication of their 2 independent probabilities. We're going to get into it a little bit deeper later, but what this means is I can just multiply these. Right? What's 1 times 1? You can ignore the 4th because it's going to be the same every time. So what's 1 times 1? What's 1 times 2? What's 1 times 1? What's 2 times 1? Right? You go from here, there, to there, then you go from here. Right? So what's 2 times 2? What's 2 times 1? 1 times 1, 1 times 2, 1 times 1. And that's your genotype ratio. 1, 2, 1, 2, 4, 2, 1, 2, 1. That's how I remember it, because I'm basically doing a factor, a multiplication of a factor. Now, phenotypically, this will look one way, and all these will look the same way. They would be yellow and round. These would be round and green. These would be wrinkled and yellow. And this one would be wrinkled and green, okay? So we have a phenotypic ratio that's different from our genotypic ratio. Our phenotypic ratio is 9 dominant to 3 hybrid to 3 hybrid to 1 recessive. Well, if we can do this by math, couldn't we do the same thing with the other one? Three yellow to 1 green, 3 round to 1 wrinkle. So how many are going to be round and green? Round and yellow, sorry, the dominant one. 9. How many are going to be round and wrinkled? 3. How many will be green and round? And how many would be green and wrinkled? 9, 3, 3, 1. So there's 16 possible combinations. Of those 16 possible combinations, they can be grouped into 4 groups phenotypically. Okay. We call it double dominant, hybrid, hybrid, double recessive. So the 9 that are double hybrid and the 1 that is double recessive look identical to the original parents because you started out with round yellow and wrinkled green. So they look just like the parent. But the other ones, the hybrids, don't look like the parents. You can also call them non-parental. They don't look like the parents. Okay. So the important thing is this observation laid the basis for the independent assortment rule. Okay. So the independent assortment rule is right there in blue. That's your definition. That's your working functional definition to memorize. Notice it says either allele. You can't use the word each here because each means every one. But this just means one of them. Either of them, either allele of one locus may be placed together or may go together with either allele of a second locus, but there is one important caveat. You have to put the word unlinked in here. Of a second unlinked locus. And we might as well dive into that right now, okay? Linked and unlinked, but we were just saying that linked didn't work. It had to be independently assorted. What's really moving during meiosis? Hopefully you read through that chapter, worked some of those problems. What's moving? When meiosis happens, what's actually separating? The chromosomes, right? So it's not just one gene on a chromosome, is there? There's lots. So what does that mean if there are 2 genes on the same chromosome and that chromosome goes to that cell? Won't those 2 genes both have to end up in that cell? Not necessarily, but what if they're really, really close by? If they're really close by, the likelihood that they end up together is higher than if them being separated, right? So what we notice is that because multiple genes are on each individual chromosome, when the chromosomes separate, the likelihood of 2 genes on the same chromosome ending up in the same cell together is higher than ending up separately. So therefore, we say that they are linked. And what happens is with inheritance is you notice that traits won't really separate. They won't really follow this rule of independent assortment. Okay, and there are some things like that. Like there are some factors of our phenotype that are associated with other factors of our phenotype very often, right? And it's tricky because sometimes they're associated because the majority of people in that group have all the same chromosomes for all the traits. Like for example, how often does blue eyes go with brown skin? Not very often. Why is that? It's not because it can't, it's because most of the people with brown skin already have brown eyes, and so most of the time, you're passing on both traits, but still independently. But they look linked, okay? But there are other traits that are linked, that are really linked, okay? Now, when we look at those linked traits, when they're on the same chromosome, what we'll notice is we won't get a 9:3 to 3:1 ratio from our F2s. What'll happen is you'll get a lot more, in this, if these were linked, what you would notice is you'd get a lot more green wrinkled ones than, they wouldn't be the fewest. They'd actually be pretty predominant. And instead, the wrinkled green and the wrinkled yellow and the round green ones would be the very fewest, okay? So it's pretty obvious when it happens. When independent assortment isn't being followed, and it's gonna be because the genes happen to be on the same chromosome, when that happens, you can see it immediately in that ratio. So instead of it being 9:3:3:1, it'll be some really wacky ratio, but what'll be in common is that if you think that the two on the end will be almost the same. The parent types will be almost the same, and the middle types would be reduced, much reduced, okay? And who knows what the actual numbers would be, but it'll just follow that trend. A whole lot of them will look like the parents, and very few of them will not look like the parents. Okay, does that make sense? And we're going to talk about linkage right this minute, but I'm just introducing it. So that's why this word has to be in here. It's important. Okay, this does not hold true for genes which in fact are linked, because they in fact do not follow independent assortment. Okay, so some people will say that linkage is non-Mendelian. And I say, quackery. Linkage is Mendelian. Okay, it follows the rule. It just follows the rule for linkage instead of for independent assortment. This is not the only rule, okay? It's the two principles. And with these two principles, you can see how everything else works. And that's why we harp on them so much. The alleles have to be separated during meiosis. If that doesn't happen, really bad things happen, okay? Now, once they're separated, the separation of an allele for one gene on one chromosome is going to be completely independent of any other gene on any other chromosome. But it's not going to be independent of its neighbors on its own chromosome, okay? Again, just like with the monohybrid crosses, he happened to do a dihybrid test cross. He said, I ought to get the same idea happening, or I get the same outcome happening. I should be able to take one of these heterozygous plants that I think is heterozygous, and I should be able to mate it to a homozygous recessive. Sorry, the book just decided that they were going to use tall in yellow and green instead of wrinkled, so whatever. But you get the idea. You have a heterozygous plant, so it's tall and yellow because of the dominance. Now it gets mated with a homozygous recessive that would be short and green, okay? This is why I hate using the lowercase letter. If you looked at that, what would little y mean? Would that make you think of green? It doesn't make me think of green. I have to work at it every time to go, that means green. Green. It's like one of those tricks that they do to you where they write the number in words in a different color. So they'll write like the number 2 in blue and ask you what the number is, and you'll say blue because it's just one of those weird things. Okay, so we get that stuck in our brain and we work with it. So what do you get? Well, it's possible to get one heterozygote. It's possible to get one homozygous recessive, and it's possible to get two different kinds of hybrids where you get a tall green and a short yellow. Okay, so you get one of each possible kind of offspring. There's only 4 different possibilities, and instead of getting 9 that are like dominant, you only get equal numbers. So for monohybrid ratios, you end up with a slight difference here. You end up with for monohybrids, then you end up with a 1-to-1 S cross ratio. Okay? This means you took a heterozygote with a dominant phenotype and you crossed it with a homozygous recessive. Okay? With the dihybrid cross, you're going to end up just doing it twice. You're going to get 1 to 1 to 1 to 1. Okay? You're doing the same thing. You're taking a heterozygote, now it's a double heterozygote with a dominant phenotype, and you're crossing it to a homozygous recessive, but for 2 traits, and you're going to get 4 phenotypes. Okay? One, and they're all going to be equal. So we say that they're equal, 1 to 1 to 1 to 1 to 1. That's equal. Okay? So look through those problems, and Thursday we'll just work some of them, okay? Because then by working them, you'll see how the phenotype and genotypes are written and how we do them, okay? I might ask somebody to come work it, so you better work it. You can look at the problems, you can say, okay, these are kind of the same. Work one of them like that, okay?